Hello and welcome. This is writing a formal laboratory report part 4. This is the last part, this is the conclusion, and it's the part I know that a lot of you have been dreading. It's really not going to be that tough though. You can do this. So this is what the scoring guide looks like for the conclusion section. There's 3 paragraphs: the introduction, the results with evidence and explanation, and the procedural errors. But to start off, we are going to be writing formally. So what does that mean? This is a form of technical writing. This is not like the writing that you're taught in most of your English classes, but this is the kind of writing that you would do for business. If you go into business, if you go into retail, if you go into science or anything like that, you're going to have to write daily reports. You're going to have to write reports of your own progress, of a group's progress, and things like that. And it will be formal. So formal means that you can't have any abbreviations. The exception to that is metric measurements. So you can use mm for millimeter, you can use cm for centimeter, that's fine. For scientific names, you can abbreviate it kind of, if you know how to abbreviate it. So the first time you write the scientific name, it must be written out completely, exactly like we talked about in class, with the genus capitalized, the species or the specific epithet lowercase, and the whole thing in italics. But the second time you do it in the same paper, you can use just the first letter of the genus capitalized, and then the full specific epithet. So you could use H. sapiens instead of Homo sapiens, and that is fine. You should write your laboratory reports, the conclusion, in past tense. So be careful about that, and maybe when you give it to somebody else to read, you can ask them to look for tense, because this is something that you have a lot of trouble with sometimes. You should sound like a young scientist presenting results, because that's who you are. Third person is preferred. I'm not going to require it in honors biology, but understand that it is required in honors chemistry, and it will be required in many of your college courses. So if you want to go ahead and get in the practice, especially if you're taking honors chemistry at the same time and you don't want to change too much and you want to get into that routine, that's fine. It is more correct. So in third person, you would say the student carried out the lab instead of I carried out the lab, or she carried out the lab. So we get to paragraph 1. First of all, you have to briefly explain what you did. So you're not going to go through the whole entire procedure, but you may say things like a time frame would be in there. We put 26 seeds in petri dishes. The petri dishes were treated as follows, and we measured germination for 3 days, or whatever you did. So briefly explain what you did. Explain how you did it. So if you used any materials that you aren't familiar with, you should explain how to use that, because your audience isn't going to understand that either. Include the correctly written scientific name. So this is where it has to be. It will probably also occur in other places in your conclusion, but it must be here because this is where I'm grading it. And the first time you have to write the whole thing out. After that, you can abbreviate it the way that I told you to on the previous slide. If you don't know the scientific name of the species that you're working with, Google it. Paragraph 2, you're going to actually say that your hypothesis was supported, or your hypothesis was refuted, or your hypothesis was inconclusive. Specifically state that. Your evidence is referred to directly by the title. So refer the readers to your data table or to your graph and say, as can be seen in the green line on the graph, the number of seeds germinated increased from day 1 to day 2, but then leveled off from there. So you want to refer directly to that. Discuss how you know what you know. Explain how you know. Hit me over the head with it. Tell me exactly how you know. Use some words from class. Explain to me what germination means. Explain to me how you measure germination. Okay, paragraph 3. These are the procedural errors. So you're going back to your procedure, reread the procedure that you wrote, and you're going to critique it. What worked and what didn't work? If you were going to do it over again, how would you rewrite that procedure? And you don't actually have to do this, so you have unlimited funds and unlimited time. How would you fix this? How would you make the results more reliable? State errors in the procedure as written. That could have been that you didn't have a real control, that you had more than one independent variable. So you put green cellophane over the dish, and you put it in the incubator, and you put another one in light with a green cellophane or a different color of cellophane. So you didn't have enough controls. Sorry. You didn't have a large enough sample size. Sample size can always be an error. You can never have too many. You need to have a bunch of seeds or a bunch of whatever you're doing. Explain when in the procedure your error occurred, and talk about what possible effect the error could have had. So how did that change the outcomes of the experiment? Make it very clear to me that you understand that. And then talk about how you could correct or improve the procedure if you were going to rewrite and rerun the experiment. Lastly, the very last part of your scoring guide refers to plagiarism. Make sure you do not copy any part of your paper from your partner or a student in another class or a previous class. Don't do it. I want to know what's in your brain. I want to know what you can do. I will care about what they can do when I'm grading theirs, not yours. Okay, you should be ready to roll. Get this done. Don't wait too long, and I know you can do a tremendous job.